All right, today we are unlocking what is basically a superpower, the ability to see the unseeable. We're going to dive deep into the invisible world of molecules and figure out how scientists can identify a substance without ever actually seeing it.
So put yourself in this spot for a second. Imagine you're a chemist. You've spent hours in the lab, you've mixed all your chemicals together, and you've made something. But what is it exactly? You can't just pop it under a regular microscope and see the individual molecules, right? So how do you know you actually made what you were trying to make?
And that quote right there nails the fundamental challenge for anyone working in chemistry. You absolutely need a rock-solid way to confirm what you've made. To get a definitive, yes, this is it answer about the structure of a molecule that's completely invisible to your own eyes.
Okay, so how in the world do we navigate this unseen molecular world? How can we possibly figure out a molecule's shape, what its bonds are, its whole structure? Well, it turns out the answer is you have to talk to the molecules. And the language you're going to use? It's light. This is where our amazing tool comes into play, spectroscopy. I mean, in the simplest terms, it's just the study of how light and matter hang out and interact with each other. But the key is this. By shining different kinds of light on a sample and just watching what happens, we can decode a ton of information about the molecules inside. Yeah, think of it just like a conversation. We send a message in using light, and the molecule sends a message back to us, usually by absorbing some of that light. And our whole job is to interpret that message. It sounds complicated, but it all boils down to one single, fundamental concept: energy. Now, this little equation here, this is the key to everything. See, molecules are kind of picky. They can only exist in very specific, defined energy levels. They won't just absorb any old photon of light you throw at them. No, they'll only absorb a photon, a little packet of light energy, if it has the exact right amount of energy to kick it up from one allowed level to the next. It's that super specific energy jump that makes all of spectroscopy work.
So let's start our investigation with our very first technique. We're going to interrogate the bonds inside a molecule using a specific kind of light, infrared light. And when you shine infrared light on a molecule, something really cool happens. The bonds, they start to vibrate.
You know, the best way to picture this is to completely stop thinking about chemical bonds as these rigid little sticks. Instead, imagine that they're tiny springs connecting the atoms. And just like a real spring, they can stretch, they can bend, and that infrared light, well, it provides the perfect little nudge of energy to get them moving.
Okay, but not all of these little molecular springs vibrate at the same speed. Just like with regular springs, a couple of things really matter. First up is bond strength. A stronger, stiffer bond, think of a triple bond, is going to vibrate at a much higher frequency. And second is the mass of the atoms involved. Bonds between lighter atoms, like hydrogen, vibrate way faster than bonds with heavier ones. And this is awesome because it gives us very specific frequencies to look for as clues.
And get this, these springs don't just move in one simple way. Oh no. They can stretch where both atoms move away from the center together. That's symmetric stretching. Or one can be moving in while the other moves out. That's asymmetric. They can also bend and wag and twist. And every single one of these unique movements absorbs its own very specific frequency of infrared light. Okay, so we've zapped the molecule, we've made its bonds vibrate, and we've gathered our evidence. So what does this evidence actually look like? Well, this is how we get something that's famously known as a molecular fingerprint. So yeah, what does it look like? I mean, the data you get from one of these machines isn't just a simple yes or no. It's this incredibly rich, detailed graph that is just loaded with all the clues we need to start cracking the case of our unknown molecule. What we get is this thing called a spectrum. Now, along the bottom, you've got this number. It's called the wave number, but really it's just a way of measuring the frequency of the light. And all those big dips, or peaks depending on how you plot it, they show us exactly where the molecule absorbed the light. So take acetone here. You see that huge sharp peak right around 1715? That is a dead giveaway. It's the classic signal for a carbon-oxygen double bond stretching. And then you see those peaks over around 2900? Those are telling us all about the CH bonds. Super useful. Now, chemists are smart about this. They basically break the spectrum up into two main areas. First, you've got the functional group region. This is where you look for the big, obvious clues. You know, the carbonyl groups, the OH bonds, the triple bonds. It gives you the broad strokes. But then, there's the fingerprint region.
Now this area looks kind of messy. It's full of all sorts of complex peaks from all that bending and stretching. But don't let that fool you, because it is so important.
And this quote just says it all perfectly. Look, lots of molecules might have, say, a carbonyl group. But the exact messy combination of all those little peaks in the fingerprint region, that is unique to one and only one molecule. So if you can match the fingerprint region from your unknown sample to a known one, boom, you've got a positive ID, case closed.
But what if we need more evidence? What if the case is a little tougher? I mean, IR spectroscopy is amazing for figuring out bonds, but it's not our only tool. So let's get a second angle on our molecule. This time, we'll use higher energy light, ultraviolet and visible light. And this technique, it doesn't look at vibrating bonds. It looks at the electrons themselves.
Here's how UV-Vis works. You basically zap a molecule with enough energy to kick an electron from its cozy home, what we call the HOMO, or highest occupied molecular orbital, all the way up to an empty, higher energy orbital, the LUMO. That energy gap between the HOMO and the LUMO, it tells us a ton about the molecule's electronic structure. And here's the really cool part. If a molecule absorbs light in the visible part of the spectrum to make that jump, well, that's why it appears colored to our eyes.
And it's important to know it's not the entire molecule that's doing this light-absorbing dance. It's a specific group of atoms inside the molecule, and that part is called a chromophore. By analyzing the exact wavelengths that this chromophore is soaking up, we get yet another layer of evidence to help us identify our molecule.
So let's step back for a second. We've now gathered evidence from a couple of different angles, right? We've interrogated the molecule's bonds using infrared light, and we've excited its electrons with UV-Vis light. We've really seen how spectroscopy gives chemists this incredible superpower, the ability to see the invisible and crack these molecular mysteries.
And here's the whole detective process laid out nice and simple. First, you start with your mystery. What is this stuff? Then you gather your first set of clues. You run an IR spectrum to find the big functional groups and, crucially, to check that unique fingerprint region. After that, you might run a UV-Vis spectrum to get clues about the electronic system. And by combining all of that data, you build an airtight case, and you can identify your molecule with total confidence.
It's pretty amazing, right? We've seen how this simple conversation between light and matter lets us identify a single type of molecule. It is such a powerful idea. And that kind of leaves us with one last big question. If we can use light to understand the chemistry happening inside a tiny flask, what else can it reveal? From figuring out what distant stars are made of, to understanding the super complex proteins inside our own bodies, what other invisible worlds are just waiting for us to unlock them?